So can you hear me okay? Yeah, okay. So first of all, thank you to the organizers for giving me the chance to present some of the work I've been doing for the last 15 years here today. So as Martha said, my name is Martine and I'm an associate professor in Phage Biology and Biocontrol Group at the University of Copenhagen. And today I'm going to talk about some published data, but also some new data that we have on these Fletcher virus phages that are infecting Campylobacter jejuni. So let's see if I can. Yeah. So first of all, why are we interested in working with these phages and this bacterium? So Campylobacter, and particularly Campylobacter jejuni, is the most common cause of bacterial foodborne gastroenteritis in humans. And it's estimated by WHO that there's around 400 to 500 million cases of campylobacteriosis occurring worldwide each year. And even though the infection is not as severe as for many other bacterial pathogens, this very large number creates a huge socio-economic burden for the society. So we know that Campylobacter is a gut commensal of many warm-blooded animals, and in particular, it's found in avian species, where it often is able to colonize to very high numbers, so around 10 to the 9 CFU per gram intestinal content, without causing any disease in the animals. And therefore, we also know that the main transmission route to human disease is associated with this colonization of the gut. And it has been confirmed or shown that the main route is contaminated chicken meat. So the contamination occurs during slaughter when the gut is kind of cut open, and then the bacterium is being transferred to the chicken meat. So there's been a lot of research going into how can we reduce the level of Campylobacter contamination in these chickens, in broilers. And one of the ways that has been a lot of studies related to is the use of phages to try to see if they can kind of lower the levels of the bacterium in the gut of particularly chickens. But there's also a lot of inconsistent results. So there has been a lot of studies using the same phage cocktail, where in one study they had up to 3.2 log reductions, and in another trial, no reduction at all. So there's a lot of inconsistency and a lot of variation. And on top of that, it has also been demonstrated that the effects seem to be transient. So there's a reduction in the beginning, and then the Campylobacter levels go up again. What has also been shown is that the phage population remain more or less stable in the gut. So the phages seem to survive very efficiently, even though you have this variant level of Campylobacter host being present. So based on these results, there is a new report coming out on different ways to kind of attack this Campylobacter problem in these broiler chickens. And there's this report by EFSA that came out in 2020 saying that the use of phages targeting Campylobacter in broiler chicken have a high probability of achieving reductions in the human cases of Campylobacteriosis. So phages are being recognized as a very promising intervention method against this pathogen. But one of the causes of uncertainty is this: how do we tackle with this variation in these results? And that has been linked to understanding the specificity of these bacteriophages, as this has been mentioned as one of the main causes of uncertainty. So that is kind of where my work comes in. So what we have been interested in our group is to try and study this initial interaction between the phage and the host bacterium. So my work has been mainly focusing on trying to, from the host side, to understand what are the phage receptors, how are they regulated, and how on the phage side do the phage recognize these receptors, and in that way try to identify these receptor binding proteins. So the phages that we are working with are called Fletcher virus, and they are this very unique group of conserved lytic phages that are infected Campylobacter jejuni. And you can see a picture of them here, that they are myvirus and they have these contractile tails. So our phage collection consists of 38 of these filaria-virus phages, and they have been isolated in Denmark from both chicken and duck. And what we know is that they are quite easily to isolate from this environment. So they coexist with their host in the same biological niche, so in the gut of these avian species. Currently, we have 19 phage genomes that have been sequenced. And what we know is that even though there are a lot of research labs out there that have isolated a lot of phages, it is very difficult to sequence them. So we developed a new method in our group that allowed us to kind of sequence this high number of genomes. And what we can see is that they are very conserved within this group, but they show very low sequence similarity to other phages. And therefore, you also have a lot of offs of unknown function. But what we do know, and what has also been shown from other people, is that they are generally dependent on capsular polysaccharides for infection. And that kind of leads me to introducing you a bit to the surface structures that are actually encoded by this bacterium. So as you can see from this picture here, Campylobacter has this nice spiral-shaped morphology, and it's highly motile due to the presence of two polar flagella that are extruding. And as I mentioned before, we have this thick layer of capsular polysaccharides that is surrounding the bacterium. But underneath, we also have a layer of lipo-oligosaccharide, also called LOS. And again, on the flagella, we also see that the flagella are highly glycosylated due to O-linked protein glycosylation. And what is important is that we know that there's a lot of variation going on in these surface structures when you look at different strains. And the variation is both due to different gene content in the loci that are responsible for producing these surface structures. But what is very important when it comes to Campylobacter is also that you have a lot of these surface structures that are being regulated by phase variable gene expression. And in Campylobacter, phase variable gene expression occurs due to the presence of N-terminal polymeric G tracts. So this is a stretch of Gs. It's normally between 7 to 11 Gs in a row. And during DNA replication, you can have mispairing of the strands, and that will kind of introduce either insertion or deletions in the stretch of Gs, and therefore you will end up having frame shift mutation. And because Campylobacter does not encode the normal mismatch repair systems that we see in many other pathogens, we have a very high frequency of these mispairing occurring, leading to a frequency of phase variation between 10 to the -3 to 10 to the -4 mutations per cell division. And this is a reversible stochastic event. And in that way, you can have a rapid switching of gene expression, so on and off, in the individual cells of a clonal population. And this means that even though you have the same genetic background within a given population, you will always have many subpopulations that are expressing different types of surface structures. So as I said, the CPS functions as a phage receptor. And even though we have this huge variation in terms of the surface structures that are produced, what has been shown is that there's one modification called the O-methylphosphoramidate structure that is found in approximately 70% of all jejuni strains. And when you look at the picture that I have illustrated here, the structures that are highlighted by this red star, they are actually phase variable surface structures due to the presence of these poly-G tracts in the transferases that are responsible for attaching the modification to the backbone sugar. And what you hopefully also can see is that these modifications are not small structures. They are actually quite bulk structures that in that way completely change the conformation of these CPS structures when they are expressed or not expressed. And what we found is that actually this MEOPN structure is a common Fletcher virus phage receptor. But what we have also seen is that phase variation can occur really rapidly, both in vitro, so in the lab, but also in the chicken, by the phase variable expression of the transferases that are responsible for producing this MAOPN receptor. But not only the receptor, also some of these other phase variable components are, when they are expressed, able to block or prevent the phage from binding. So one of the examples that we have studied is a phage called F336, which infects the strain called 11168. And you can see here that when you have the MAOPN attached to a GAL-F backbone structure, that forms the phage receptor, the phage is able to infect. But if we have a switch in the tract, in the transferase responsible for putting on the phage receptor, then the phage will no longer be able to infect. Okay, so that was kind of summing up a bit what is going on the host side. So we were, of course, very interested to figure out how do the phages cope with this very variable surface structure that you have occurring all the time, plus the ability of the bacterium to generate phage resistance so rapidly. And what we discovered by sequencing 18 or 19 of our phages is that they actually encode up to 4 different receptor binding proteins. And as you can see here, most of the phages encode all 4, but we also have examples of some phages that only encode a few of these. But what was even more interesting for us was to discover that 2 of these receptor binding proteins actually concern poly-G tracts, so mimicking what we observe in the bacterial host. And this suggests that also the receptor binding proteins are phase variably expressed in the phage. What we've also noticed for a long time is that when you perform a plaque assay using these phages, what we see is we often have this, or we in every case have this dual plaque phenotype. So we define this as turbid plaques and clear plaques. So while the turbid plaques are always produced at the same frequency, we see that the clear plaques are often variably produced. So not always will we end up having the same number of clear plaques occurring on a plate. And what we now know is that expression of RBP2 is associated with this clear plaque formation. And what you can see here is an example of the same phage, but propagated to a new stock. And what I hope that you will be able to see is that in this particular stock, you have a much higher frequency of clear plaques occurring. And we now know that this is due to the phase variable expression of RBP2 associated with the number or with the presence of these hypermutable poly-G tracts. So regardless of when you propagate the phage from a clear plaque or a turbid plaque, you will always end up having these dual plaque morphologies, but the frequency of the clear plaque formation can vary. What we also discovered is that RBP2 expression actually ensures phage infection when this MAOPN receptor is not expressed. And I hope that you can see that it's only the clear plaque formation that we have occurring on this receptor mutant. So this kind of illustrates that both the host and the phage are actually able to perform phase variation by the same mechanism. And that kind of creates population dynamics that promotes coexistence of both phage and host in the shared niche, so in the chicken gut. And what we know is that RBP1 is responsible for binding to the MAOPN receptor. And as I mentioned before, RBP2 is able to then bind to the host when the MAOPN receptor is not being expressed, but we don't know what the receptor is yet. So, but again, this is a way for lytic phages to kind of coexist with the host in the same niche. But we also have an unusual phage in our collection that is independent in terms of the CPS for the host recognition. So it's still a Fletcher virus. It's highly conserved genetically when we compare it to the other CPS-dependent Fletcher virus phages. So we only have approximately 9 open reading frames that are different for this phage compared to the other. But what we have shown is that it's actually relying on motile flagella for infection. And here you can see that it's attaching to the flagella through the tail fibers. And what we have also seen is that if you paralyze the flagella, the phage is still able to infect its host, but with a much lower efficiency. And we therefore have a hypothesis that this phage, F341, uses the flagella rotation to kind of travel along the flagella filament to interact with the secondary receptor at the bacterial body. So of course, knowing the region where these receptor binding proteins are encoded in the Fletcher virus phages, we looked into this region for the F341 phage, and what we could see is that it actually does encode another receptor binding proteins. So it has an N-terminal that is kind of showing partial similarity to the RBP1 that we found in the CPS-dependent phages. But when we then look at the C-terminal, we can see similarity to a tail fiber that is actually encoded in cryptic prophages that we see in many of our CG2 genomes. So suggesting that this RBP has kind of evolved by recombination event between the lytic phages and these cryptic prophage elements. To kind of confirm that indeed this RBP functions as a receptor binding proteins, we raised polyclonal antibodies and performed a plaque assay, so a spot assay. And what you can see here is that when we add these antibodies, when we mix them together with the phage prior to phage infection, we are able to block infection completely. And also when we then use these antibodies in transmission electron microscopy, we can see that they are actually binding to these distal tail fibers of the phage. What we also found was that indeed this phage actually does rely on a secondary receptor, not only the flagella, because if we have a mutant where we delete the outer part of this LOS surface structure, we are able to prevent the phage infection completely. So in that way, we now know that the flagella might be the primary receptor, but the LOS is now the secondary receptor of this F341 phage. Yeah, so just to sum up, the Fletcher virus are a unique group of these genetically highly conserved lytic phages that are infecting specifically Campylobacter jejuni. So now that we are able to sequence them, we are becoming more and more aware of how unique they are and what mechanisms they actually use to kind of interact with their host. And we have discovered that they actually mimic the bacterial host by encoding these hypermutable poly-G tracts that promote phase variable expression of 2 out of these 4 receptor binding proteins. And that this phase variable expression creates these phenotypically diverse phage populations, including the subpopulation that then is able to infect the host in the absence of the common MAOPN receptor. And also that phase variation of both host surface structures, but also the phage RBPs actually create these stable but fluctuating population dynamics that can kind of promote coexistence of both lytic phages and their host in the shared niche, the chicken gut. And finally, what we recently discovered is that these cryptic prophages that we have in many of the Campylobacter jejuni genomes actually may represent a genetic pool from where these conserved viruses are actually able to acquire completely new traits. And with that, I would like to acknowledge, of course, my group, but particularly my former master student, Line, who did a lot of the work with the F341 phage, and also a PhD student, Amir, who worked with me on isolating and sequencing these phages. And of course, thank you for listening.
I'm always worried I'm going to be too loud when I hold this mic up. Hi, my name is Emma. That was an excellent talk. I was wondering, for one, have you heard of any of these types of phages that are temperate, or are they strictly lytic, or just your 19 are strictly lytic?
So all these are strictly lytic.
And then my other question was, in these phages, when you were doing whole genome sequencing, did you notice in a higher number of proteins related to homologous recombination, or are they relying more on the host for the shuffling of their adsorption proteins?
Yeah, so we do actually have a recombinase just located upstream the RBP region. So we suspect that that might have a role in the swapping. But what was also very surprising was that when we look at all the sequences that are available for these phages, all the RBPs seem to be quite conserved, also in phages isolated from other countries. So we would have expected a lot more diversity, at least in the receptor binding regions, because the surface structures of Campylobacter are so diverse. So we are still kind of interested in seeing if that will hold when hopefully more sequences of more phages kind of come.
I'll hope with you.
Yeah.
Hi, thank you for the nice talk. I'm Dibyendu, and I wanted to ask about these turbid plaques. So in general, what they are formed by, and in your specific case, have you looked at it further, what's causing the turbid plaques? You talked about RBP2 as the clear plaque formation reason.
Yes, so normally when you have these turbid plaques, it's been associated with lysogeny of temperate phages. So when you have a resistant population occurring, and what we believe is that it's simply also a resistant population, but not due to lysogeny, but due to this very rapid phase variable way of becoming phage resistant. So that is what we see with these turbid plaques. And that could be linked, for example, to turning off the expression of the MAOPN receptor, which would induce resistance. But what we also know is that you can also gain resistance by switching the expression of other of these phase variable CPS surface structures. So there are several ways for the bacteria to become very rapid phage resistance. And we think this is what we see with the turbid plaque formation.
So it's just like variation, heterogeneity in the bacterial population, they're responsible for that?
Yes.
Okay. Thank you.
Thank you. That was an exquisite talk. Really enjoy that. Thank you. Just a quick question on, so if you're to look forward now with all this resistance and kind of shuttling back and forth between sensitivity and resistance, where does this leave phage therapy in the context of protecting, our humans from Campylobacter infection?
Yeah. So what we kind of advocate is that first of all, it's important when you want to have a phage stock that you ensure that you have a stock that produces a high frequency of these clear plaques, so you know they are able to infect despite the receptor is not present. And what we also say is that the timing is crucial. You should be able to add the phages just before, for instance, the birds are slaughtered, because you don't want the population being able to kind of overcome the phage attack, so to say. So timing is crucial. And then also looking into what you actually have in your phage stocks.
This might seem like a strange addition, but is there any kind of, you will say, companion bacteria that you can use to support, we'll say, suppression of the Campylobacter in addition to the phage therapy? So you have this kind of community competition for available growth space.
Yeah, I know there are also a lot of studies where they consider adding different probiotics, so other kind of things to kind of see if that could inhibit or compete for the same niche. But what we know is that Campylobacter is so specialized in colonizing a very specific niche of the gut. So it's very efficient in doing that. And there's not a lot of competition for that niche, at least not what we know of right now. But now with the possibility to do microbiome analysis and so on, people are becoming more and more aware that maybe this is also something that we should look more into.
Excellent. Thank you very much.
Yeah.
I think in the interest of time, let's thank Martine once again for a brilliant talk. And